I'm Lynne Boddy. I'm Professor of Mycology or Fungal Biology at Cardiff University, where I've been since about 1983, I think, so a very, very long time. I teach and I research into fungi, particularly into wood decay fungi. Well, when I was an undergraduate, I was interested in any aspect of ecology, and so I suppose that when I graduated, I could have done it. I would have been happy to do anything, so maybe marine ecology, plant ecology, any sort of ecology. And it just so happened that there was a job advertised on the ecology of wood decomposition, and I thought, oh, that sounds interesting. So I applied for the job and I got it, and the rest, as they say, is history. And this particular job was to look at amounts of wood falling in forests and how much was on the forest floor and the rates of decomposition. So that's important for nutrient release. And at the time, I wasn't doing anything on fungi, but of course I obviously very quickly realised, as everybody else already knew, that fungi are really, really important in decomposition. So the next thing that I wanted to do was to work on the fungal communities in wood. And also, I was working on wood decay on the forest floor, but I very quickly realised that most wood decay starts in the canopy. Very much decomposition can occur while wood is attached to the tree. And so I wanted to work on communities in attached branches. And so with somebody at the University of Bath, Alan Rayner, we applied for a NERC fellowship, and I got that, and I started working in that area then and carried on from there. Fungi are absolutely crucial to our existence because they're crucial to the functioning of all of the terrestrial ecosystems on our planet. And that's for two main reasons. One is that about 90% of plants have fungi associated with their roots in a mutualistic pairing called mycorrhizal fungi. And the fungi absorb water and nutrients from the soil or whatever they're growing on, and they pass this to the plant in return for sugars that the plant has made in photosynthesis. And in the natural world, without these fungi, plants, including trees, really would not be able to establish and do well. So that's one main reason why fungi are absolutely crucial to our planet. And of course, indeed, all plants have fungi as endophytes within them, which also very often confer advantages to a plant as well. The second main reason is that fungi are the major decomposers on the planet. Fungi are the garbage disposal agents of the natural world. Some of them, and it is a relatively narrow range of them, have the ability to break down wood, to break down lignocellulose. And this is because they produce these very fine filaments which can find their way into solid, bulky objects like wood, and because they have the enzymic ability to break down these very complicated molecules. Very few organisms can do this. So they can break down wood. And that's important not just because if it weren't for fungi we'd be up to our armpits in dead stuff, but because within dead stuff there's a vast amount of nutrients locked within, and fungi, by breaking down dead organic matter, trees, leaf litter, whatever, they release the nutrients that are in there, which can then be sequestered by plants via mycorrhizal fungi, for example, and used for continued plant growth. Without them, the planet would soon become relatively barren because nutrients would be just completely sequestered in dead, recalcitrant material. In the early 1980s, and indeed before, people really thought that trees were a sterile environment. And I suppose people probably thought that about human beings too. They didn't realise that we had a microbiome with fungi and bacteria inside our guts, etc. And people didn't really realise that either. And it became clear very quickly that wood decay in the canopy started just very suddenly, just like that. Almost 1 minute you'd see branch which was fully functional, and then very quickly you'd see that part of it, or perhaps all of it, was decomposing. And we watched some branches and we saw that decay developed in less than one growing season. And you could find very large individuals of fungi in these branches. And we thought, well, this is pretty amazing. How are they doing this so very rapidly, perhaps rather more quickly than you'd expect them even to grow in the field? And we started to think carefully about this and came up with the idea that to get these huge decay columns in such a short time, really the fungi had to be there already. And one scenario you could imagine was that if in a column of sapwood in a branch, if there were lots of little propagules sparsely dispersed, and they were the same genotype, the same individual, the same genetic makeup, if they could be sat there all the time, they couldn't grow because functional sapwood is full of water, because obviously this is what sapwood's all about, is transporting water from the roots to the shoots. In those conditions, basidiomycete fungi really, on the whole, don't cope well, so they wouldn't be growing out. But then if you imagine that maybe a root was damaged, or some of the leaves were removed, and that sap stream stopped, the wood began to dry out, and then those little propagules could effectively germinate, mycelia could grow out from them, and as they were identical, when they met, they would fuse and become one individual. And so this was our hypothesis, that those fungi which start the decay process are already there, but they can't act because it's high water content environment, and they just sat there effectively minding their own business. Only when that wood begins to dry do they develop. So that was the working hypothesis. This was really rather against the grain at the time because, as I mentioned, people thought that this was a sterile environment. So we're saying some rather radical things. We're saying that in fact trees have lots of fungi already in them, not necessarily doing much. Now, of course, nowadays we fully accept that because everybody accepts that there are endophytes. All plants have endophytes. Endophyte literally meaning within plant. So in the leaves and the roots and shoots all over the place, there are fungi. We now know that. So we had this hypothesis anyway, that these fungi were there. We then had to test that hypothesis. And we did it at the time because there were no snazzy molecular tools around then in the early 1980s. What we did was we chopped down branches, beech branches it happened to be, from the canopy that they looked to us fully functional, fully healthy, chopped them into different lengths, covered the ends with plastic, and then revealed certain amounts of area by making holes in there. The more area it was revealed, the more rapidly they dried down. No area revealed, they wouldn't dry down at all. And we set them to drying down, and then after different amounts of time, we chopped them up through the middle and had a look to see where the fungi developed. No contaminants could have gone in through the ends, we made sure of that, and sure enough, in the Goldilocks condition, where the conditions were just right, fungi began to develop. And we showed by that approach that fungi were there, which was, really not appreciated at all at the time. So that was one of the first really exciting findings. What we know is that in attached branches of different species, the early decay communities are often different. So oak have their own set of fungi, a dozen or so fungi which develop early on. Beech have a more or less different set. Ash has a different set still. But with the molecular techniques, we've showed that the fungi from all these three tree species are actually very often present in all of the tree species, even though only some of them develop a particular set in beech, a particular set in ash, a particular set in oak. And it turns out that when you do the chopping up branches experiment and drying down at different rates, if you change the temperature, the gaseous regime, dry them at different rates, you get different fungi developing. So it seems that the conditions determine from that little mixed pot of fungi that are in there, which ones actually develop. So the step on from that is that we know our climate is changing. So the implication is probably the fungi which are the early decay community on various tree species will probably change. And indeed, there's some evidence for this already. We know from long-term fruiting records that on elder, Sambucus, in the 1950s and beforehand in the UK, the main fungus that developed early on was the ear fungus Auricularia auriculojudae. This carried on for a long time, up into the 1970s, and then suddenly other fungi began to be spotted on elder as well. And then more and more and more. But interestingly, on beech, after about 1980, suddenly we were finding Auricularia on beech. So it almost looked as if it was switching its host from elder to beech. But actually what's probably happening is it's in both of these tree species anyway, but the conditions have changed. We know conditions have changed since the late 1970s, early 1980s, and this is probably a climate change effect, and I'm sure we will be seeing that much more in all sorts of tree species and with all sorts of fungi, not just those early decayers in the canopy. It was really exciting when we first realised that there were fungi already present in wood, in what had always been thought until that time to be a sterile environment. That was very exciting. Another really exciting finding was when we realised that wood decay fungi, when they grow across the forest floor and they find new resources, when we found that they actually respond very dramatically to those new resources. And they do this in a very sort of animal-like way. So a fungal mycelium, that's the main part of the fungus, will grow out from a block of wood symmetrically. Maybe it encounters a new woody resource and it starts to colonize that. It recognizes that it's found a new source of food and sends messages. We don't know what the messages are to the rest of the mycelium, which is effectively saying this is the way to go, this is a successful search pathway. And then the mycelium, which is away from that new resource, starts to be reabsorbed back into the original lump of wood, and then those nutrients are reused. And so the mycelium changes where it's going. And if you sort of map these sort of changes, they look very much like termite trails that you'll see when termites have found a source of food. So fungi are really very dynamic and they really respond dramatically to new resources. So that's another exciting thing. And then one of my favourite lines of research has always been the way that fungi interact with each other, the way that wood decay fungi behave when they encounter another wood decay fungus. And they are very aggressive. They fight. When fungi meet, they fight. And the outcomes can be deadlock or replacement. And if they're deadlock, then the fungal community doesn't alter. And you can see where fungi are fighting. When you chop across any rotten piece of wood, you'll see these little dark lines called interaction zone lines. They're not always dark coloured, sometimes they're bright coloured, sometimes they're orange with some fungi. It depends on the fungus. And those lines represent where fungi have met each other. It can be fungi of the same species or fungi of different species. They're rather like us in that we like to put fences or hedges or walls around our homes and gardens, and they do the same, and that's defending their territory. And they then have access to all of the territory, maybe a long individual decay column, and they can use the nutrients in there at their leisure, but they just have to defend on the outside. Now, as I say, some are more aggressive, and they can expand their territory and take over from other ones, and the communities will change. So working on these sort of things has been really rather fun. And over the years, we've done all sorts of experiments, looked how climate affects the outcomes and the sort of chemicals that they produce, which bring about these interactions. So it's really rather like chemical warfare. They produce lots of volatile chemicals, perhaps a little bit like trench warfare in the First World War, all sorts of different ways of doing things. Most of our work has just been done in the laboratory with two fungi. Imagine you've got two blocks of wood here, one with one fungus, another with another. Put them together, watch them fight. Or sometimes on trays of soil, we let them grow out and fight. But of course, in the real world, you don't usually just get two fungi fighting each other. There's 3 or 4 or 5. And fungi can be very big, so they can be fighting on all sorts of fronts. So recently, we've tried a slightly more complicated situation in which You remember those toys that you used to get called the Rubik's Cubes that used to drive me quite mad with the different coloured blocks? And if you imagine each of those blocks is a different fungus, we started making Rubik's Cubes with our earliest ones with just 3 different fungi, and we put them together in different arrangements. And one arrangement might be just some sort of random arrangement. Another arrangement might be a wall of one fungus, which has got the same amount of volume as in the other block, which is just separated all over the place. And we discovered that when a weak fungus is present as a wall, it very often can defend its territory very well against much more aggressive fungi. When it's in the Rubik's Cube just dotted all over the place, it's completely replaced. And the wall sort of experiment is a rather nice mimic of the decay columns that you get in branches or in large trunks. So things in the real world are very much more complicated than our initial experiments in the lab. So that was very exciting when we find, you change the complexity and you change the answers that you get, if you like.